Okay, I just wanted to show you the slide that we left off on. This is the part 2 lecture for chapter 3 on important biological macromolecules. I just wanted to show you in this picture that everywhere that you see a point or a bend, that's just understood to be a carbon atom. So I drew the carbon atoms in for you in red. So what you're looking at is a hydrocarbon chain. Some of the hydrogens are not shown. Like, for example, this carbon would also be bonded to two hydrogens. And we don't draw all the hydrogens because it's messy. Because, chemists know that carbon makes 4 bonds. So it's assumed that the other atoms that are bonded to the carbon that are not included, that are not shown in the structure, that they are hydrogen atoms. But anyway, this just shows you the hydrocarbon chain is bent, and here's your carboxylic acid group on the end. That makes it a fatty acid. Another example of lipids are waxes. We find waxes in nature covering the surface of leaves, covering birds' feathers, and earwax, of course, is an example of a wax. So waxes are examples of lipids. They're made up of long fatty acid chains esterified or bonded to long chain alcohols. Sorry. And phospholipids are the backbone of the plasma membrane or cell membrane. Okay. So a phospholipid contains a phosphate group along with the glycerol molecule of the lipid. So here's your phosphate group and the glycerol, and both of these together comprise the hydrophilic head. Hydrophilic means water loving. So the heads are soluble in water. The heads are polar and soluble in water. And then the hydrophobic tails are fatty acids. They can be saturated or they can be unsaturated. The percentage of unsaturated fatty acids determines how fluid the plasma membrane is. If you have a high percentage of unsaturated fatty acids, the plasma membrane is more fluid. And if you have a higher percentage of saturated, then it's more solid. But the tails are hydrophobic, so they are nonpolar and insoluble in water. And the way they're arranged in plasma membranes is that they form a phospholipid bilayer or a double layer of phospholipids, where the tails face inward because they are nonpolar and hydrophobic, and then the heads face outward because the extracellular fluid is going to be water-based. So there's typically water outside and inside of the cell. So this would represent the water outside the cell, and then the water-based fluid inside the cell. We call that the cytoplasm. So the heads are exposed to the water because they are polar and hydrophilic. Another example of lipids are steroids. Steroids always start out as cholesterol. And cholesterol is also found in the plasma membrane. Cholesterol affects the fluid nature of the plasma membrane. And cholesterol is the precursor of steroid hormones such as testosterone. Testosterone is the primary androgen or male sex hormone, and then there's estrogen and progesterone, cortisol. So any steroid hormones are start out as cholesterol, and then they are converted into the particular steroid hormone. Okay, there's a practice question in here. I was getting kind of nervous because this is usually at the end. Why are lipids necessary for cell membranes or plasma membranes? And let's see, well, phospholipids are necessary because they're hydrophilic on the outside and hydrophobic on the inside. But the cell membrane doesn't need to be a solid. it needs to be something that materials can move through. And nonpolar molecules like oxygen and carbon dioxide gas, which are exchanged across our plasma membranes during cellular respiration. We breathe in the oxygen, we breathe out the carbon dioxide, but the oxygen and carbon dioxide travel through our blood and they travel into and out of our cells. Oxygen enters our tissues or our cells, and carbon dioxide is built up as a byproduct. So carbon dioxide leaves our tissue cells. But both of those are nonpolar. So anything nonpolar is soluble in a lipid. So they can simply pass through the phospholipid bilayer. So if we go back here to the phospholipid bilayer, if oxygen is going to come into the cell, it just goes right straight in between those phospholipid molecules. And the lipid structure makes the plasma membrane fluid. Makes it a fluid structure instead of a solid structure. And that's very important for the function of the plasma membrane or cell membrane. Okay, now we're on to our third group of macromolecules called carbohydrates. This is the primary energy source for our cells. And some carbohydrates called polysaccharides provide structural support to plant cells, fungi, and arthropods that have an exoskeleton, like insects and shrimp. Carbohydrates are divided into three groups. They're divided into monosaccharides, disaccharides, and polysaccharides. And mono means one sugar, di means two sugars, and poly means many sugars. So this is an example of some monosaccharide, meaning simple sugars or one sugar. Our blood sugar is glucose, which is not in this. Here it is. I'm sorry. Our blood sugar is glucose, and this shows you the structural formula for glucose. The chemical formula for glucose, which you will need to know, is C6H12O6. And that is the same chemical formula for glucose and for fructose, but they're just structurally a little different. The glyceraldehyde is a 3-carbon sugar, so we call that triose, a triose sugar. The ribose sugar, there are 2 ribose sugars. There's one called ribose and one called deoxyribose, and they are the sugars found in DNA and RNA, our genetic material. So ribose sugars have 5 carbons. They're pentose, so they have 5 carbons. Sorry about that line. And then at the top, you can see the difference between an aldose and a ketose sugar. Let me move my face out of the way so you can see that. An aldose sugar has a C double bonded to an O. On the end of the molecule, and then a ketose sugar has a C double bonded to an O in the middle of the molecule. So if you look down at the bottom, all of the sugars that are shown here, glyceraldehyde, ribose, and glucose, are aldose sugars. And monosaccharides can either be linear, like you saw here, or they can form rings. The ribose sugars form a pentagon or a 5-sided ring, and the glucose sugars form a hexagon or a 6-sided ring. Disaccharides are, can't find a good place for my head. Maybe I should just take it out. But disaccharides are when 2 monosaccharides are linked together by a glycosidic bond, which is a covalent bond. Sorry. I don't know why I keep doing that. But this is called a glycosidic bond, is the covalent bond that links together 2 monosaccharides. So in this case, what you're looking at is glucose loses an OH, fructose loses an H, the atom in red, and that forms water. And then that leaves a place where the two molecules are able to be linked together, because that leaves a bond kind of open. And they bond together at the oxygen that's left, and that is called a glycosidic bond. And what is produced as a byproduct is water. So really the chemical reaction that forms sucrose, the disaccharide sucrose, is glucose plus fructose forms sucrose plus water. And here's some other common disaccharides. We already said sucrose, but lactose that's found in milk is a combination of glucose and galactose. And then maltose, or malt sugar, is a combination of two glucose molecules. You find maltose in, like if you ever order a milkshake and you order it as you order a malt, they put malt powder in it. That's malt sugar. And like the candy, the chocolate candy called Whoppers, has malted milk on the inside. You may have had those before. And another example is malt liquor. So maltose is not as well known, but it's definitely a sugar. It's definitely a disaccharide, and it's composed of two glucose molecules. Now, polysaccharides are composed of many glucose molecules, or many monosaccharides. So like with proteins, we learned that the polymer is the protein and the monomer is the amino acid. With carbohydrates, the polymer is the polysaccharide, and the monomer of polysaccharides are monosaccharides. Okay, two examples of polysaccharide are starch and glycogen. Starch is the storage form of glucose in plants. We find a lot of starch in plant roots like potatoes and carrots, beets, rutabagas, turnips. And glycogen is the storage form of glucose in animals. So what happens is when you eat a meal, and it probably contains carbohydrates, those carbohydrates are broken down into glucose. So any sugars or any complex carbs that you eat, like bread or pasta, is broken down into glucose. And that glucose is dissolved in your blood, and it raises your blood sugar. So your liver, because of the insulin produced by your pancreas, the hormone insulin, insulin causes your liver to take individual glucose molecules from your blood. Glucose is shaped like a hexagon. So it causes your liver to bond individual glucose molecules together, and that forms glycogen. So glycogen is the storage form of glucose in animals. So glycogen is the storage form in animals, and starch is the storage form in plants. Starch is also chains of glucose. Now, if your blood sugar is low, normally what you do is eat. But if you don't eat, if you skip a meal or something like that, your liver can release glucose back into your blood to raise your blood sugar. It can break down this glycogen and release the individual glucose molecules back into your blood. And so that reaction can be reversed. Another type of polysaccharide is found in plant cell walls, and it's called cellulose. And this is just to show you that cellulose fibers are parallel, but there's still chains of glucose. So cellulose is still a polysaccharide, and it forms from chains of glucose. And this is amylose, which is a type of starch, and amylopectin. Both of these are constituents of starch. Glycogen is similar to starch as far as its chemical structure.